Implantation. It is the process of embedding of the conceptus, as known as your blastocyst, in the endometrium of the uterus in normal events. It can occur at other sites also. The blastocyst lies free in the uterus for about 2 days. Then zona pellucida gradually degenerates and disappears. About 6 days after fertilization, the blastocyst attaches to the endometrial epithelium at embryonic pole. Blastocyst at this stage consist of embryoblast or inner cell mass and outer trophoblast. Trophoblast soon differentiates into the inner cytotrophoblast and the outer syncytiotrophoblast. The latter consists of multinucleated cytoplasm and the cytoplasmic mass. It produces proteolytic substances. They erode the maternal tissues. It has finger-like processes that extend into the endometrium and help in the process of implantation of blastocyst. By the end of first week, the blastocyst is superficially implanted. Critical events in implantation is timely disappearance of the zona pellucida. If it dissolves early, then blastocyst may get implanted in fallopian tube itself, leading to tubal pregnancy. On the other hand, if it dissolves late, implantation may occur close to the internal os, leading to placenta previa. There will be the driving force. The secretions from the syncytiotrophoblast serve as a driving force for the blastocyst to invade into the maternal tissue. The uncontrolled invasion of the maternal tissue is, however, prevented by the decidua. It counters the action of syncytiotrophoblast. Here we'll find some normal and abnormal sites of the implantation. So various sites of implantation that you can see in this image. Normally implantation occurs in the upper part of the posterior wall of the uterus close to the fundus. Tubal implantation usually occurs in the ampulla, but it can occur in the infundibulum or in isthmus also. Tubal pregnancy may burst open and the conceptus may be secondarily implanted in the abdomen, being called abdominal pregnancy. Implantation close to internal os leads to placenta previa, and this is associated with the maniac of antepartum hemorrhage. Then the events of the second week starts. So events in the second week, mainly in the second week, two things happen. That is, the embryoblast, or you can say the inner cell mass, becomes bilaminar embryonic disc and comprises of an epiblast and hypoblast. Trophoblast that differentiates into an inner cytotrophoblast and outer as a syncytiotrophoblast. Here you'll find two cavities that develops here, that is the amniotic cavity along the epiblastic face and the yolk sac on the hypoblastic side. During the second week, the process of implantation completes. This is possible due to erosive syncytiotrophoblast. It has been described as ingestive, invasive, and digestive. But this invasive and erosive nature of syncytiotrophoblast needs to be countered, and this is achieved by surrounding the endometrial stromal cells. They acquire lipid and glycogen, become polyhedral and are now called the decidual cells. This conversion of stromal cells into decidual cells is called the decidual reaction. The trophoblast at this stage is differentiated into mitotically active cytotrophoblast that proliferates rapidly, and as it does so, the cells move outward. These cells lose their cell wall, giving rise to syncytium, that is, protoplasmic mass with multiple nuclei without cellular delineation. This forms the syncytiotrophoblast. With this, the trophoblast now has inner cytotrophoblast and outer syncytiotrophoblast. The fate of this embryoblast, when the inner cell mass or the embryoblast organizes it into flat disk comprising of an epiblast presenting the columnar cells and a hypoblast presenting the cuboidal cells. So, a bilaminar embryonic disc is formed. Formation of two cavities also occurs concurrently, that is, an amniotic cavity and the yolk sac. Though these cavities are formed in the second week, they develop and have differentiative role in subsequent part of the gestational period. It will be appropriate to describe them at this stage. First we'll see the amnion. It forms membranous amniotic sac that surrounds the embryo. The amniotic cavity appears as a small space between the embryoblast and invading trophoblast. Its floor is formed by the epiblast and the roof is formed by the amnioblast cells. The amnioblast cells are derived by the delamination of the cytotrophoblast. The embryonic cavity contains the amniotic fluid. So, from where this amniotic fluid comes? What is the source of this amniotic fluid? This amniotic fluid secreted by the amniotic cells initially derived from the maternal blood by transport across amnion from the decidua parietalis and intervillous space of the placenta. Fetal urine makes sizable contribution towards the term, about 500 ml per day. Volume of the amniotic fluid at 10 weeks it will be 30 ml, at 20 weeks it will be 350 ml, and at 37 weeks it will be 1000 ml. Less than 400 ml in the third trimester is called oligohydramnios. More than 2000 ml is referred to as a polyhydramnios. The large amount of amniotic fluid passes through the amnion into the maternal blood. The fetus swallows amniotic fluid. It is absorbed by the gastrointestinal tract, passes in the blood and the waste products in it pass across the placental membrane into maternal blood. Fetal kidneys excrete some part of it into the amniotic fluid pool. What is the significance of this amniotic fluid? Amniotic fluid provides buoyancy effect, permitting the symmetrical growth of the embryo. It protects the embryo against injuries received by mother on abdomen. Thermoregulation of the fetal body temperature. It prevents adherence of the amnion to the embryo. It provides aquatic medium around, enabling the fetus to move freely, aiding the musculoskeletal development. Then we will see the yolk sac. In the second week of the development, two cavities appear in relation to the bilaminar embryonic disc. Amnion on the dorsal side and the yolk sac on the ventral side. It is called primary or primitive yolk sac. Its roof is formed by the hypoblast to be replaced by endoderm. It is surrounded by the extraembryonic mesoderm, which is formed by the delamination of the cytotrophoblast. In this mesoderm develops an extraembryonic xylem. With this, a part of the primary yolk sac is separated from it. This forms a exocoelomic cyst, while rest of it forms the secondary yolk sac. During the fourth week, there is folding of the embryonic disc. Within this, a part of yolk sac is incorporated within the embryo. Part of the yolk sac in the head folds forms the foregut. The the part within the tail fold forms the hindgut, and the intervening part forms the midgut. The part of the yolk sac that lies outside is connected to the midgut by yolk stalk. What is the significance of this yolk sac? During the second and the third week, it is responsible for transfer of nutrients before uteroplacental circulation is established. During the fourth week, its dorsal part is incorporated within the embryo to form the foregut, midgut, and hindgut. So it is giving rise to lining of trachea, bronchi, lungs, gastrointestinal tracts, and the associated glands. Primordial germ cells are formed in the wall of the yolk sac, then they migrate to the developing gonad and form the germ cells, that is, spermatogonia and oogonia. Hemopoietic activity is also done by this yolk sac. The formation of the blood cells occurs in the wall of the yolk sac in the third week. Allantois develops as a diverticulum from the caudal end of this yolk sac and projects into the connecting stalk. What is this allantois? The word allantois is derived from a Greek word alas, means sausage. It appears as a finger-like outpouching from the caudal part of the yolk sac around day 16. It projects into connecting stalk. Initially, it is continuous with the developing urinary bladder. Soon, it constricts and becomes a thick cord-like structure called the urachus. It connects the apex of the bladder to the umbilicus in subsequent life. It gets obliterated to form the median umbilical ligament in the postnatal life. So, what is this significance of this allantois? In human beings, allantois remains small and is involved in early blood formation. It is also associated with the development of the urinary bladder. In reptiles, birds, and some mammals during the embryonic life, allantois has a respiratory function and also stores the urine. Allantois subsequently thickens to form the urachus and may exhibit some anomalies. So here the chorion and chorionic villi also develops. So chorion, that is consists of extraembryonic somatopleuric mesoderm and the overlying trophoblast. The extraembryonic mesoderm is formed by the delamination of the cytotrophoblast cells. It surrounds the amnion and primary oxa. Soon the cavities appear in the extraembryonic mesoderm. They fuse to form the larger cavity, the extraembryonic xylem. With this, the extraembryonic mesoderm is split into inner splanchnopleuric layer covering the yolk sac and the outer somatopleuric layer under the trophoblast. This outer somatopleuric layer, along with the overlying cytotrophoblast and syncytiotrophoblast is called the chorion. Towards the end of the second week, the cytotrophoblast starts proliferating, and these proliferation form the column-like structures, the primary chorionic villus. Subsequently, it is invaded by the mesoderm, is known as your somatic layer of the extraembryonic mesoderm, to be called the secondary chorionic villus. Soon, within the mesodermal core, fetal capillaries appear, which is referred to as a tertiary chorionic villus. With the development of the chorion, the extraembryonic xylem is now called the chorionic sac, in which the embryo, the amnion, and the yolk sac are suspended. Till the end of the 8th week, villi cover the entire surface of the chorion. The villi associated with the decidua capsularis become compressed. Their blood supply is reduced and they degenerate. So they just produce an avascular bare area called the chorion leaf. Levis stands for the smooth. So chorion levy, that is formed when the avascular bare area starts. Concurrently, villi that abut against the decidua basalis grow rapidly and branch profusely, giving rise to the chorion frondosum. Frondose, that stands for leafy. The chorion frondosum forms the fetal component of the placenta. At this stage, the embryo is in the form of bilaminar disc with columnar cells forming the epiblast and cuboidal cells forming the hypoblast. However, in a localized area at one end of the embryonic disc, the cuboidal cells of the hypoblast become columnar, and this forms the precaudal plate. This forms the future site of the mouth and is an important organizer in the head region. This latter forms the buccopharyngeal or oropharyngeal membrane. So here we have completed the events in the second week. In next class we will study the events in the third week. So thanks for watching.